Hello biology students, and welcome to video 3 of the organic chemistry unit. So today we're going to be talking all about lipids. And in fact, this video is going to be one of 2 videos just about lipids. So lipids are non-polar. So remember, that means that they share all their electrons equally in their bonds. And they're hydrophobic. And if we break this word down a little bit, phobic means having a fear of, and hydro meaning water. So this word literally means afraid of water. So what this means is that lipids don't mix well with water. And based on what we learned last unit, that makes sense. Non-polar molecules are not going to be able to form hydrogen bonds, so they're not going to be able to hydrogen bond with water, and therefore are not going to mix nicely. Okay. So just like carbohydrates, lipids are made up of carbon, hydrogen, and oxygen, so the exact same 3 atoms. However, the difference is they have less oxygen than carbohydrates do. So if you remember the monosaccharide, the monomer of a carb, the formula was C6H12O6. So if this molecule were going to be a lipid, maybe it only would have 2 oxygens, for example. I'm just making that up. But you can see we'll have less oxygen than the same carbohydrate. So there are 4 main types of lipids: triglycerides, phospholipids, waxes, and steroids. This video is going to be all about triglycerides. And finally, the function of lipids, they're used for long-term energy storage. So they have twice the amount of energy as a polysaccharide. So remember we talked about how in humans, the storage polysaccharide is glycogen? Well, fat, or lipids, can store twice as much energy as glycogen can, and can store it for a longer period of time. Lipids or fats are also used for insulation, for protection. If you were to take one of your organs out of your body, like your kidney, and you were to hold your kidney in your hand and look at it, it would be covered in fat, and that's because that fat is meant to protect your organ. So lipids are used for protection. And then finally, lipids are also used for waterproofing. You can think of maybe like a leaf on a plant has a waxy coat to it, and that's a lipid, and it's helping to prevent water leaving the leaf when it shouldn't, or entering the leaf when it shouldn't as well. Okay. So the first main type of lipid we're going to talk about, and in fact the only one we're going to look at in this video, are triglycerides. And these are your fats. What you think of as fats and oils are triglycerides. So technically, a triglyceride is actually a polymer. Okay, so it's made up of many pieces, and the monomers, or the pieces that make up this polymer, the triglyceride, are 1 glycerol molecule and 3 fatty acids. So those are your 4 monomers, the 3 fatty acids and the 1 glycerol. Okay, so here I have a picture. So this is glycerol right here. For sure, you should definitely know this structure. So you can see I just have a 3-carbon chain with 3 hydroxyl groups. And what happens when I'm building a triglyceride is just like when I'm building any polymer, I'm going to do dehydration synthesis. So my other monomer that I'm going to use is my fatty acid here. So what I'm going to do in dehydration synthesis, I'm going to take away a water from the 2 monomers, and then this fatty acid will join to this glycerol. But remember, if we look back at the definition of a triglyceride, they're made up of 3 fatty acids. So what I would do is I would attach a second triglyceride via dehydration synthesis right here with this hydroxyl group, and then a third one right here with this hydroxyl group. So if we look at the next slide, what a triglyceride will ultimately look like is that glycerol molecule will do 3 rounds of dehydration synthesis, and we'll link 3 fatty acids. So here my 3 fatty acids are now all linked to my glycerol molecule. And this is a triglyceride. So whenever you see that kind of E-shaped, and maybe part of the E might be a little bent, right, that should be a big clue to you that we're looking at a triglyceride. Remember I said you have to know the structures. You have to be able to recognize all the structures of these molecules for this unit. Okay, so it turns out there are different types of triglycerides. And you know this. If you look at a food label of something you're eating, you see things like saturated fats, unsaturated fats, monounsaturated fats, right? You've seen those words before. And remember we said 2 slides ago that triglycerides are fats. So we're going to look at those different types and figure out what the difference is. Okay, so a saturated fat or a saturated triglyceride have all carbon-carbon single bonds. So that's what I mean by this. If you have to write in carbon-carbon single bonds, do that so you can remember. Okay? And what what this means is that because every carbon on this fatty acid has a single bond, the single bonds all form a nice straight shape. So this means all the fatty acids are nice and straight. There's no kinks, there's no bends. So because they're all straight, they can all pack in really closely together. And because they can all pack closely together, they end up being solid at room temperature. So you might remember from previous science classes that if molecules are really close together, you get a solid. If they're a little bit further apart, you get a liquid. And if they're very far apart, you get a gas. So because these fats are all nice and straight, they can pack in close together and become solid. So saturated triglycerides are things like animal fats, like butter, the fat you find on red meat, like steak. Those are all saturated because they're solid at room temperature, and that's because they can pack in nice and close together. Another type of triglyceride or fat is an unsaturated fatty acid, or an unsaturated triglyceride. And unsaturated triglycerides have at least one carbon-carbon double bond. Okay, so you could write it in double if you're not sure you're going to remember that. And what this happens, wherever there's a double bond, we end up getting a kink or a bend in the molecule. So I know it's kind of hard to see, but this bond right here between this carbon and this carbon is a double bond. And what that does is it makes my carbon chain bend. So it goes single, 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 nice and straight. And then there's a double bond, and that introduces a bend in the molecule. So what this does is now my triglycerides can't pack as closely together, and this makes them liquid at room temperature because the molecules are not as close. So these are things like oils or the fats you find in nuts and plants. And it turns out these fats are a lot healthier for you. And I want you to think about why for a second. So I'm going to be silent for a minute so you can think. Okay. So it turns out unsaturated fats are healthier for you because when those fats are in your bloodstream, they stay liquid. And if they're staying liquid, they're not going to form plaque or fat buildup in your arteries. You can think saturated fats or saturated triglycerides are solid at room temperature, so they're going to be more solid while they're in your blood vessels, and they might form plaque or buildup or blockages because they're solid. Okay, so just because I know you're going to see these words, a mono unsaturated triglyceride has one carbon-carbon double bond. Poly unsaturated triglyceride means you have one or more, so one or more kinks. And you can kind of think the more kinks or the more bends you have, the more liquid we're going to be, so the better it will be for you because it will not clog your arteries. So the next thing I would like to talk to you about is the difference in the names of a saturated triglyceride and an unsaturated triglyceride. Because we know saturated, they all have single bonds, so they're straight and they can pack in tightly and be solid at room temperature. And unsaturated triglycerides have that double bond that makes a kink and makes them pack less tightly and be liquid at room temperature, but it can kind of be hard to remember which one is which. So I want you to look at the word saturated. So what this means in English is it means that something is full. So for example, when you get out of the shower and use a towel, if the towel was saturated, it means the towel will be full of water. So in this case, at the molecular level, if we say something is saturated, we mean it's full of hydrogens. So in a saturated triglyceride or a saturated fatty acid, the carbons are each bonded to the maximum number of hydrogen atoms they possibly could be. And because they're full of hydrogen atoms, they don't have any empty bonds, so they all form single bonds between the carbons. On the other hand, in an unsaturated fatty acid or an unsaturated triglyceride, the carbons are not holding their maximum number of hydrogens. So you can see this top one is only holding 2 hydrogens, where it could in theory be holding 3. And this one's only holding 1, where it could in theory be holding 2. So because the carbons have open bonds, they end up forming double bonds with each other, and that introduces the kink, which makes them pack less tightly and be liquid at room temperature. So hopefully that helps you remember which one is which. So finally, we're going to come to the worst triglyceride. And I know you've heard of this. You've heard the phrase trans fat. So what this is, when we have a nice, healthy, unsaturated fatty acid, so something with a double bond, right? So we're holding less hydrogen because we're unsaturated. And what the industry does is they break this double bond, okay? And what they do in the food processing industry is they add more hydrogens to it. And this is called hydrogenation. You might have heard of partially hydrogenated oils, and you might know these are bad for you. And that's because they're taking a healthy fat, one that has a kink in it, so it is nice and liquid at room temperature, and they're adding hydrogens to make them more solid. And this might be because they don't want their product to be runny, they want it to be more solid, they want it to stay together more easily. They want it to have some kind of property that a saturated fat does. But basically all they're doing is taking a nice healthy fat and making it more unhealthy. So that makes it the worst kind of fat or the worst kind of triglyceride for you. So these do a lot of bad things for you. They raise your bad cholesterol levels, they lower your good cholesterol levels, and overall, you want to try to avoid trans fats wherever possible. Okay, so then finally, to round up our discussion of triglycerides, I want to talk to you about triglyceride metabolism, or in other words, how your body deals with triglycerides when you eat them. So we know we have the glycerol molecule, right? So here's my glycerol. Okay, hydrogen, hydrogen, hydrogen. Okay, and then we know we have the 3 fatty acids, and I'm not going to draw them all out, but there's my triglyceride. So if your body's going to break this down, the first thing it's going to do is it's going to break it into subunits. So it's going to break it into the glycerol, and then it's going to break it into the 3 fatty acids. Okay, and then these subunits are going to be used for cell respiration, which is a process by which the body takes food and turns it into energy. And then any that's not used for energy via cell respiration is going to go being stored in the adipose cells. These are fat cells. And here they're going to sit. So that's it. That's video 3 all about triglycerides. And I'll see you in the next video where we'll finish out lipids.